Hello everyone, welcome back to this course in Neuroscience, De Natura Cerebri. In this episode, we're going to dive into the world of neuroanatomy. This will lay the foundation for all the following episodes. Now, let's get started. In this episode, we're first going to learn about some terms. This includes terms for indicating locations and terms for bunches of neurons and bundles of axons. Then we're going to have an overview of parts of the nervous system. Here is the list of structures that we will cover today. Now, here are some terms for indicating location. First, this is a relatively straightforward set of terms, left and right. Basically, left is to the left and right is to the right. Now here is another set of terms, medial and lateral. Medial means closer to the midline of your body. We're all bilaterally symmetric organisms, which means that we have a midline, a line of symmetry. For example, in this little mouse, closer to the midline, we have medial structures. Further away from the midline, we have lateral structures. Similarly, in a human body, we have the midline here. These are the medial structures near this midline, and the lateral structures there. We have yet another set of terms for the sides of the body. That is ipsilateral and contralateral. Ipsi means the same, and contra means different. So my right hand and my right foot are ipsilateral, but my right hand and my left foot are contralateral. Now we're going to talk about up, down, and front and back. The most straightforward set of terms are anterior and posterior. Anterior is front and posterior is back. Superior and inferior. Superior is upward while inferior is downward. Rostral and caudal. This is more tricky. In a mouse like this one, rostral means closer to the mouth or the nose, while caudal means towards the tail. However, in humans, we have a turn of the neuraxis, which is the axis of our neural system. In human bodies, we can divide the human body into two parts. In the brain, rostral means towards the front and caudal means towards the back. While in the spinal cord, or to say in most of our body, rostral means upward while caudal means downward. A related set of terms is dorsal and ventral. Dorsal means towards the back, while ventral means towards the belly. Here, in a mouse, ventral is downward and dorsal is backward. In a human body, we still divide it into two parts. In the brain, ventral means downward, while dorsal means upward. In the rest of the body, ventral means front and dorsal means back. And finally, we have another set of terms for indicating the transections of the brain. That is to say, if we cut the brain, how do we describe how do we cut it? This is a sagittal section. Sagittal comes from the word meaning arrow in Latin. So it's like this. A mid-sagittal section is the one sagittal section that is corresponding to the midline. That is straight at the middle, while other sagittal sections may be a little bit off to the side. This is a horizontal section. It's horizontally cut. And finally, this is a coronal section. It's like wearing a crown. Finally, there are some things that we must notice when we are looking at transections. That is, we have a difference between brain sections directly cut from the brain and neuroimaging pictures. On the left, we have a brain that is cut. On the right, we have an MRI scan of the brain. These are upside down and left side right. When we go into the MRI machine, we lie down and gets transported into it. Therefore, the MRI machine looks at us from the feet to the head, while usually we look from the head to the feet. And that is one of the reasons for the difference between these conventions. Now let's get to the terms for bunches and bundles. For bunches of neurons, we have cortex, which means the layer of neuron at the surface of the brain. Nucleus, which are clearly distinguishable group of neurons inside the brain. Substantia, it is a group of neurons with less distinct borders also inside the brain. Locus, a small but well-defined group of neurons inside the brain. And ganglion, a collection of neurons in the peripheral nervous system, which means outside the brain and the spinal cord. And bundles of axons. We have bundle, which means a group of axons running together, but not necessarily in the same direction. Tract, which means a group of axons with the same origin and destination that are running together. Capsule. Capsule is a larger bunch of axons inside the brain. We have the internal capsule and the external capsule. The common point is that they connect the cerebrum, the brain, with the brainstem. Commissure. Commissures are one type of bundles of axons that cross the midline. They connect symmetric structures on the two sides of the brain. Decussation is the other type of bundles that cross the midline. Here, one side goes down to the other side, and this side goes down to the other side as well, forming a cross-like structure. Lemniscus. Lemniscus means a ribbon-like long group of axons. Later, we will meet the medial and lateral lemniscus. And finally, nerve. This is the term used to describe a group of axons in the peripheral nervous system. There are certain exceptions, and we will come upon that later in this episode. Finally, we have a contrast between neuron soma and axons. You might have heard of this: gray matter and white matter. Gray matter means a region where there are abundant soma of neurons, and white matter means a region where there are abundant myelinated axons. They are named like this because white matter looks white and gray matter looks gray in a fresh brain segment. Gray matters look gray because the somas are usually gray, while white matters look white because of the fatty myelin sheaths. Now it's time to have an overview of the nervous system. The nervous system consists of the central nervous system and the peripheral nervous system. The central nervous system, abbreviated CNS, is here. It includes two parts, the brain and the spinal cord. The brain consists of three parts: the prosencephalon, which is also called the forebrain. That is because pro means forward and encephalon means the brain, so we can just call it the forebrain. The mesencephalon, which is also called the midbrain. And the rhombencephalon, which is also called the hindbrain. The forebrain consists of two major parts. One is called telencephalon, and the other is diencephalon. Diencephalon is further subdivided into the thalamus and the hypothalamus. In the hindbrain, we also have two segments. One is the metencephalon, and the other is the myelencephalon. The myelencephalon is also called the medulla oblongata, or usually just simply the medulla. The metencephalon is further divided into the pons and the cerebellum. That's the brief structure for the central nervous system. In the peripheral nervous system, we have cranial nerves, which go out from the central nervous system in the brain, and the spinal nerves, which leave the central nervous system in the spinal cord. We have a subsystem called the autonomous nervous system, the ANS. This is a subsystem of the peripheral nervous system. It consists of some of the cranial nerves and some of the spinal nerves. Finally, there are some other structures that are not nervous tissue but are very important to us. They are the ventricular system, the meninges, and the blood vessels. Let's start looking at all of them one by one. The first and foremost is the first part of the prosencephalon, the telencephalon. The telencephalon is the major part of our cerebrum. At the surface of our cerebrum, we have the cerebral cortex. We can divide the cerebrum into two hemispheres, one left and one right. On the cerebral cortex, we have many sulci and gyri. A sulcus is a groove in the brain surface, like this, and the gyrus is a bulge on the brain surface. These help us increase the surface area and accommodate more neurons, and that makes us more intelligent. In fact, humans have more sulci and gyri than most of the mammals. And finally, we have some deeper grooves. They're called the fissures. This is the longitudinal fissure, and this is the lateral fissure. The cortex can be divided into 4 lobes. They are the frontal lobe, the parietal lobe, the occipital lobe, and the temporal lobe. They are named according to the bone that covers them. So the frontal lobe is under the frontal bone, etc. They each have different functions, and we will come upon them later one by one in later episodes. There is a 5th and hidden lobe called the insula lobe. If you pull apart the lateral fissure, you can see the insular lobe inside. It's sometimes also just called the insula. The cortex also can be divided into many cortical areas that carry out different functions. For example, we have the motor area, the sensory areas, and areas for higher functions. A scientist named Brodmann has divided the cerebral cortex into 43 areas, and we often name the cortical areas according to the Brodmann areas. For example, the primary visual cortex is Brodmann area 17. The telencephalon also has some subcortical structures. A very important one is the corpus callosum. The corpus callosum is a very large group of bundles that cross the left and right sides of the brain. That is to say, it is kind of like a giant commissure. Here is another group of structures called the basal ganglia. This term is sometimes considered obsolete or inappropriate, but just for convenience, we will still use this term in this course. The basal ganglia consists of the striatum, which further consists of the caudate nucleus and the putamen, and the pallidum, and some other smaller structures. We also have the limbic system, which is also sometimes considered an obsolete form. Its definition also varies across different books or different people. So here we can say that the limbic system consists of the hippocampus, the amygdala, etc. The diencephalon is the second part of the prosencephalon. It stands between the 2 hemispheres of the telencephalon, so in the middle here. The diencephalon consists of the thalamus, which is very important for information relay. The hypothalamus, which is the grandmaster for the internal secretion system, and the pituitary gland. Here we have the mesencephalon, the midbrain. There are many important structures in this small midbrain. Let's talk about one here. It's called the substantia nigra. This name simply means a substantia, a group of neurons without a very clear boundary that look black in a fresh brain section. It is related to Parkinson's disease. In a Parkinson's patient, the substantia nigra neurons die out. See here, the black color fades, and that causes some motor dysfunctions. There are many other structures, but we will not cover them in detail here. In the rhombencephalon, we first come upon the metencephalon, which consists of the pons and the cerebellum. Let's zoom in on the cerebellum. The cerebellum consists of deep cerebellar nuclei, which are deep inside the cerebellum, and the cerebellar cortex, which is on the surface of the cerebellum. The cerebellar cortex consists of folia, which are the little stripe-like structures that you see on the pictures on the right. Folias form lobules. There are 10 lobules in the cerebellum. And finally, the cerebellum also consists of 2 hemispheres, the left and the right. In the middle of the 2 hemispheres, we have the vermis. This is the word that means worm. It indeed looks like an earthworm. And finally, this is the last part of the brain, the myelencephalon, also called the medulla oblongata. There are many small nuclei inside the medulla, and they are also very vital for our vital functions. We will talk about them in later episodes. The spinal cord. The spinal cord is a thin stripe of nervous tissue that lies within our vertebral column, within our bones. This is a transection of the spinal cord. In the spinal cord, we have ventral roots, which are nerves that leave the ventral side of the spinal cord. We have dorsal roots, which are nerves that enter the spinal cord on the dorsal side. And finally, we have dorsal root ganglia. This is a group of somas of the neurons of the dorsal roots. Now let's get to the peripheral nervous system. In the peripheral nervous system, we have the cranial nerves and the spinal nerves. There are 12 pairs of cranial nerves. They are numbered 1 through 12. The numbering is according to the most rostral to the most caudal. The first two pair of nerves, CN1 and CN2, belong to the CNS. They are the olfactory nerve and the optic nerve. We say that they belong to the CNS because, firstly, they develop with the brain instead of with the other nerves in the peripheral. And second, they are myelinated by oligodendrocytes, which is a feature of CNS structures. In contrast, cranial nerves 3 through 12 belong to the PNS. They are myelinated by Schwann cells instead of oligodendrocytes. Spinal nerves. There are many pairs of spinal nerves, usually 30 or more. Each pair of spinal nerves enters or exits the spinal cord in the gaps of the vertebral segments. Each nerve combines the nerve roots from the ventral and dorsal roots. The autonomous nervous system, as we mentioned before, is a combination of certain spinal nerves and certain cranial nerves. It has two subsystems. One is the sympathetic division. This is also called the fight or flight division, and the parasympathetic division, which is also sometimes called the rest and digest division. They have very diffuse control of many of the organs in our body, and they help regulate our vital responses and vital functions. The sympathetic and parasympathetic division have opposing functions. When one is activated, the other is repressed. The NS involves both cranial and spinal nerves. One notable structure is the sympathetic chain alongside the spinal column. Here is the sympathetic chain. It has many ganglia consisting of the soma of the neurons in the ANS. And finally, we have the enteric nervous system. It consists of nervous tissue in the walls of our intestine. So it's somewhat like a brain in your guts. The enteric nervous system directly controls the peristalsis of our intestines. Now, we're about to exit the nervous system and look at some surrounding structures. The ventricular system. The ventricular system is a system that is filled with cerebrospinal fluid. Cerebrospinal fluid is a kind of fluid that provides support as well as nutrition for surrounding brain tissue. The ventricular system consists of many ventricles. From the rostral to the caudal, we first have the lateral ventricles. There are two lateral ventricles, one on both sides of the brain. Then going down, we have the third ventricle, which is in between and a little bit inferior to the lateral ventricles. It is located between the two halves of thalamus, between the diencephalon. It is located between the two halves of the diencephalon. Further going down, we have the cerebral aqueduct, which is inside the mesencephalon or the midbrain. Then the fourth ventricle. This is between the pons and the cerebellum. And finally, going downward, we go to the spinal canal, which is in the middle of the spinal cord. Eventually, the CSF, the cerebrospinal fluid, exits the ventricular system and enters the meninges. Eventually, the cerebrospinal fluid, CSF, exits the ventricles and enters the space in the meninges. It is then absorbed into the blood. By the way, the CSF are produced inside the ventricles from the arteries. Talking about the meninges are three layers of material that surround our brain and our spinal cord. The three layers are dura mater, arachnoid mater, and pia mater. Mater basically means mother in Latin. So these three layers are like a mother who protects your brain very carefully. Dura mater is the hard mother. Arachnoid mater is the spider-like mother. The name is due to the spider web-like structures under the arachnoid mater. That is to say, there are spider-like connective tissue in the subarachnoid space. And the third layer, the pia mater, is the soft mother. It follows the grooves, or the gyri, of the brain. Inside the meninges, we can find cerebrospinal fluid and veins. The cerebrospinal fluid is absorbed into the blood at certain points in the meninges. And talking about blood vessels, we have arteries and veins in the brain to supply nutrition as well as to get rid of metabolic waste. We can find arteries inside the brain tissue and in the meninges. Around the arteries, there is a blood-brain barrier. We have mentioned this when we were talking about astrocytes. The astrocytes helps to form the blood-brain barrier, which prevents certain substances that are harmful to the brain to enter the brain from the arteries. The veins are also found inside the brain tissues and in the meninges. That's all for this episode. In this episode, we have learned about the terminology for indicating location, as well as to describe bunches of neurons and bundles of axons. Then we have had a very panoramic overview of parts of the nervous system. Here are some recommended resources for studying neuroanatomy. In the appendix of Chapter 7 for Neuroscience Exploring the Brain, we can find many exercises that are really good for practicing neuroanatomy and memorizing all the structures. In the second row, we can find a 3D brain model, which is also very recommended for getting a sense about the 3D structure of the brain. And UBC Neuroanatomy is a very good resource for looking at sections of the brain that are clearly labeled. Some other resources are UBC Clinical Neuroanatomy, the Internet Pathology Lab, and the Allen Brain Map. Thank you for watching this episode. This episode intends to provide a map for the brain so that you can refer to this map in later episodes. We have learned about the structures, and in the next episode, we are going to talk about the development of these structures. How did the brain form from one single fertilized egg? See you in the next episode.